Okay, welcome to another episode of the Common Babbler. It's a podcast to discuss science pedagogy and discussion of scientific questions at a level of undergraduate classroom. And my guest today is Professor Christian Griesinger. Professor Christian Griesinger is director of Max Planck Institute of Multidisciplinary Sciences, Göttingen, Germany. And I think previously the institute used to be Max Planck Institute for Biophysical Chemistry. Professor Gessinger is an expert in NMR spectroscopy, and he has this is one of the very reputed groups internationally of NMR spectroscopy, and their recent work into neurodegenerative diseases. And Professor Gessinger has won a number of awards for his throughout his career for his contributions, and currently. I think one of the molecules that are, or some of the molecules, so a family of molecules that is designed in his group are really at a very advanced or reasonably advanced stage of clinical trials, which are showing excellent results, and it's a very promising development. So we are thrilled to have Professor Gresinger. I'm Kalyan Chakrabarty. I'm an associate professor of biological science and chemistry at Kriya University in the School of Interwoven Arts and Sciences. And Professor Gresinger, welcome.
Thank you very much for having me here.
This is, in fact, your second visit, and you have been here 2 years before. Do you notice any changes?
Well, I mean, the room is as cold as it was last time. So this time I'm not in a t-shirt and shorts, but together with the sweater. I mean, more buildings have come up, and I also was told that the number of students has increased by a factor of 3, and the faculty increased, I think, by 50% or so. So, I mean, this time, I don't remember last time that I had also the chance to talk to students at length. So yesterday evening, I had a very nice discussion with some 7, 8 students or so, and I was really impressed with the quality of their education. They were between 1 and 4 in the first and in the fourth year. And I mean, the discussion was really at a high level. It was very broad, and I must say that it seems that you really attract here very good and able students, which are very motivated to pursue life in science, at least in the coming years, mainly PhD, master's thesis, and PhD. And I really congratulate you as one of the members of the team here of this success. Yeah, and I mean, of course, yesterday you showed me the boxes within NMR spectrometers. 2 years ago there were no boxes, and so it is to be expected that you are now then also up for running experiments yourself, and that of course is absolutely essential that the equipment is there, that you can do the work for what you were hired, right? Because I mean, at least in German universities, we are still trying to follow the Humboldt ideal, which means that research and teaching are done by the same people, and only a good teacher is also somebody who has direct contact with research or is pursuing research. And so that should be integrated. And of course, for that, you need to have the equipment that now is at least there in the boxes and will be unpacked in the coming weeks.
Thank you. Thank you for that's very encouraging. I was curious about what you said about Humboldt, the philosophy, and it's my understanding that the current structure of higher academics, like having the postdoc or PhD, the way it's designed, is kind of the German system that historically it was this kind of long phase of scholarship with a single instructor, the way PhD is designed. Is that did that originate in Germany?
Whether that I don't know,
Okay.
because I mean, Germany has some old universities, and but the oldest I think in Europe was Bologna, and how they did it with a PhD, I cannot really tell. Um, so single advisor, I mean, that is still somehow the rule. Still, I mean, there's now more and more, and especially in Göttingen, they have these thesis advisory committees where 3 people are responsible, normally the direct supervisor, and most of the time one has one. There are cases in which one has 2, for example, because one is going back and forth between 2 places. We have, for example, a partner lab in Rosario, and if a person enrolls in such a PhD, then this person will be in Göttingen as well as in Rosario, and then the thesis advisory board also has at least one person, has exactly one person from Rosario, if the main time is spent in Göttingen and vice versa. So these advisory boards are common, and I understand they're also planned or implemented here. I think this is, this is a pretty new development. I think this is taken actually from the United States, where this had been the case already before, and it certainly is kind of a safeguard for the students, that they are not only at the mercy of their supervisor, but that some other people are also looking on the progress and maybe also on the direction and so on, so that it is not just the decision of the PhD student and the supervisor in which direction the thesis goes, or when one considers it to be kind of finalizing and so on. So that's a good development for sure. And the other good development, of course, of these thesis advisory boards, which are pretty new in Göttingen, something like maybe 20 years old or so, is that one understands more what the supervisor, the direct supervisor, is doing. And if one finds it interesting or so and wants to start a collaboration, one can do it during the time that the PhD student is still there. Yeah? While in former times, one was informed more or less about the topic of the thesis only when the thesis was over. Yeah? And then, of course, it is impossible to do anything. Yeah? So it also fosters the exchange on campus while the projects are still being executed, and I think this is one of the greatest inventions. Whoever did it in academia. Yeah.
Okay. We recently in Indian education system, very recently there is a shift in the policy, in the sense that the undergraduation has become 4 years instead of 3 years, and the masters were previously 2 years. But currently, I think the proposal is that after 4 years of undergraduate education, one can directly sign up for a PhD. Or I don't know if this will ever will be practiced, that have a 1 year masters. Uh, I know I should have known what is the system in Germany
In Germany, it's 3 years bachelor and then 2 years master, including the master thesis, which is normally half a year. So in principle, after 5 years, then one is a master and then can do a PhD. We also have these integrated master PhD programs in the form of so-called international Max Planck research schools. And there are 3 of that in Göttingen, one for biophysics, one for molecular biology, and one for neuroscience, which take bachelor students, and then they have a very, very intensive year with lectures and courses and also lab rotations, 3 lab rotations, which are going on for 8 weeks or so. And after that, then there's a test, and if they pass the test with a good mark, with an A, then they can go directly to a PhD, or decide they want to do a master and then a PhD. If one has a B, I think one has to do a master, and if one has a C, maybe it is not possible to make a master. So most of the students choose to do a master, which in a way offsets a little bit this attraction of having an integrated master PhD. But that is due to the fact that they think, okay, I mean, if I have a master, I have a master, and if something goes wrong with my PhD or so, I don't even have a master. So that's probably the the argument behind. But it's possible in principle then for especially for systems in which the bachelor is only 3 years, that these people apply to this, and then in principle they are, I mean, really on a fast track. They could then start their PhD after 4 years of study,
Right.
would be really kind of the turbo way to a PhD. And I mean, that this option is there is nice, because I mean some people are maybe bored by listening to lectures and doing lab work or so that is kind of reproducing experiments that people did already, and maybe they have done as pupils already so many experiments in their kitchen lab that they don't need to do that anymore. So that option exists, but still kind of bound to these integrative courses. And these international Max Planck schools are all over Germany in different topics. And I mean, it's possible certainly to apply with the 4-year or the 3-year bachelor.
I will ask, I heard the fascinating talk about the connection between Parkinson's disease and alpha-synuclein. But before that, I wanted to ask you more about the very adventurous cycling trip that you were planning.
Yeah, I mean, I was in 2018 together with a colleague from NIH doing a trip in Kathmandu Valley, and we were hoping to see the Mount Everest from there, but it was always so dusty or polluted or whatever, the air, that we did not see it, despite the fact we were in a hotel which was called Mystic Mountain Hotel.
Okay.
And the Mystic Mountain was the Mount Everest. But still, although it rained in the night, the next morning we still couldn't see it. So then our tour guide showed us some pictures of people who he said were less well conditioned as we were, and so this idea arose to do that. So yeah, we are still in the planning phase of this, and maybe it's going to happen this September, October, which should be a good time, not a rainy season there. And let's see how many people volunteer to join. And let's see. I mean, it will certainly be a thing that one does only once in a lifetime. It's pretty challenging altitude-wise, from 4.2, I think, in Lhasa to 5.2 at the base camp, which one passes by,
Okay.
and then going down all the way to 800 meters in Kathmandu.
Is this the Annapurna base camp? That's the base
No,
camp?
it's a Mount Everest base camp.
Mount Everest
Yeah,
base camp. Yeah. Okay.
At least to my knowledge, yeah.
Okay.
No, I mean, according to the tour guide's schedule, it's the Mount Everest.
It's a Mount Everest.
And there we should see it then,
Right.
finally. I mean, if one doesn't see it from the base camp, oh my God. I hope one doesn't have to climb it in order to see it.
So crossing the Himalayas on bicycle and Tibet to Nepal.
Yeah, well, I mean, it's possible. I mean, our guide showed that this is possible, and I guess it is easier than to climb Mount Everest, which also a lot of people are doing. So, yeah.
Well, all the best.
Thank you very much.
That's an incredible adventure.
I mean, it will be the challenge, of course, is the altitude, especially in the first days, and then also biking up, and so one has to be. I mean, it's difficult to forecast whether there's any altitude sickness. So I think one just has to experiment with one's own body and see whether it happens or doesn't happen.
These are, I'm guessing, that mountain terrain bikes that, but are you getting any special gear, or this is the...
I don't know. I mean, it's all in the hands of the guide.
Okay.
As far as I understand, these are mainly roads.
Okay.
So I guess it's more road bike.
Okay.
Because one doesn't really have to go over stones or specifically steep things. But I guess it will be a street bike with enough gear, because at certain places it is a little bit steep, but I really do not know.
Okay.
I will bring my helmet, although probably I don't even have to bring it. So
Right.
it's yeah, and also I mean the luggage will be transported in a car.
Okay.
The idea apparently is to camp
Mhm.
and to have a cook that
Okay.
then makes the meals. Although, I mean, the question, of course, is whether in the altitude one has the same appetite as
Right.
maybe one is fighting against nausea all the time, and
Right.
then the best meal will not appeal.
Right. Right. It's truly adventure of a lifetime.
Let's see. Yeah.
Heard your talk and the recent developments on treatment of alpha-synuclein or potential treatment of
Mm.
alpha-synuclein that are coming out
Yeah.
of the laboratory. And I know that we had this discussion before 2 years ago, and I think you mentioned at that time that there is some, you were waiting for some results which you think that are kind of was a bottleneck for a long time. Um, is that the oligomers that are now being
I mean, we
proposed?
we have this molecule which we are now pursuing in the clinic since many years, and this is in principle a development that the company that we founded is pushing, and that one has to fight with regulatory offices and so on to put all the files together in order to do that. I'm not operationally involved in that. That is really the company work, and mainly the work of my colleague Armin Giese, who is fully now in this company. And we teamed up, and that I think had happened already then with company Teva, which is also has a great clinical team, and so I think it's I mean, this development is just going, and one has to just be patient and wait what comes out. I mean, from a scientific perspective, what was a little bit unclear is the mode of action.
Right.
And I mean, that in principle is a question of the structures, and so we have just now a month ago or so published a high resolution structure of an oligomer which has the right properties that one would expect from such an oligomer, namely being toxic to cells and also making calcium ions to flow through a membrane, and of damaging also mitochondria, and that can be rescued with the alpha-38B on this specific intermediate. So it has the right properties, namely of being toxic and then being detoxified. And so we are now trying to understand the structural changes there, and with the structure of an alpha-synuclein oligomer, and the fact that we see that the alpha-38B also binds to this oligomer and modifies it structurally, which we don't quite understand yet what exactly is happening. But there is a structural change, and that is in principle what we were expecting, namely that the structure somehow changes, and that then leads to different properties of this oligomer. So that's in principle a new development since the last visit here, and it's. I mean, we see interesting effects which are online or in line with the kind of expectations that such a oligomer and modified oligomer should have, and we try to do that not only with alpha-synuclein but also with A-beta and IPP in order to also try to understand why one molecule works in mouse models where different proteins are aggregating. And I mean, the results that we have now make me kind of hopeful, or maybe even confident, that we can find out in the coming couple of years or so more about this kind of detoxification mechanism, and probably also on more than just one molecule, namely the alpha-synuclein. And maybe on the next visit, then I can present more about these specific mechanisms. This is still a hope, but at least some observations that were in the lecture today could be made, and I mean, they make me really hopeful and confident that maybe when the result for the clinical trial is there, we also have some results of kind of maybe explaining at least the structural biology, and maybe then even in case of success explaining the efficacy of the molecule in humans.
in your next visit, definitely would like to hear about that, along with also the Tibet to Nepal cycle trip.
Okay. I
Professor
for sure will
Griesinger
take pictures.
I know that a lot of biophysicist groups were trying to characterize the oligomer. That was really the sort of the
Mm.
holy grail. And these oligomers are extrememely difficult because they were transient and it was like an invisible state. Uh, so I think was this using the lipid membrane and some sort of assembling the oligomer on the
Yeah.
lipid membrane,
Yeah.
was that the
I mean,
technical
there
breakthrough
was a
to?
PhD student, Leif Anton Schmidt, who experimented with aggregation, and I don't exactly know why he switched to the lipids. I mean, this was one of the um, um, conditions that he was trying out, and then by careful observation, he saw that oligomers were populated, while if he studied the aggregation in the absence of membranes, then this was not seen, at least to the same amount. And so that then led to the discovery that one can really distinguish different intermediates on pathway to the fibrils. And then with this preparation method, it was possible to identify identify this intermediate one, separated from monomers, which is essential in order to that it is stable for some time. Normally, the aggregation goes within a few hours or so to completion to the fibrils. But if one then depletes this thing from the monomer, then the aggregation is stopped, and then the sample is stable for something like 2 weeks or so, and that enabled us to do the structural investigation, because something that is stable for a few hours only, we could not analyze. And so that, in principle, this preparation was the breakthrough. And Mo Kyung Han, a postdoc from the lab, she's now working with A-beta and also with membranes, and she also found a method to observe intermediates on the pathway to the fibrils. They have different spectra, so that must mean that they have different structure than the fibril. And also, if one then adds the 138B to this oligomer, as well as to the fibrils, one sees chemical shift changes, and that also indicates that, first of all, binding is happening, and second, that probably there's also some structural changes happening. And so that's probably the next in line where we can hopefully then maybe even compare between the alpha-synuclein oligomer and the A-beta oligomer. What is the effect of the small molecule on this? And A-beta, of course, is in a way easier because it is smaller. It's A-beta 40, and so it has only 40 amino acids, while alpha-synuclein has 140 amino acids. And so spectroscopically, it's more complicated than to assign as compared to the A-beta 40. And IAPP is another molecule that we look at, which is even smaller, 37 amino acids, but it's a little bit more complicated to handle it. And so hopefully we can also get intermediates or on the pathway to the fibrils, and then do similar things in order to get this potential commonalities of how the molecule works. But for example, it's in the case of alpha-synuclein, when we aggregate it in the presence of lipids and the alpha-synuclein, we find the same number of fibrils or same amount of fibrils as in the absence. While, for example, for A-beta, this is not the case. There's really a reduction of the fibrils formed, and that we don't observe for alpha-synuclein. So it could also be that different mechanisms play. I mean, if one just reduces the number of fibrils, and maybe then we have also indications regarding the oligomers, also has smaller population of the oligomers, I mean, that would already be an explanation. That there is, I mean, just less toxic species and then less toxicity. But this is still speculation, and we have to see what really comes out. But at least we can have a preparations method for these species, and so we can find out what they do, and yeah, that's probably all we can hope for.
So one thing I was thinking that these are the solid-state NMR methods where it's a snapshot of the molecule, drug molecule is bound to the protein assembly. But of course, NMR and especially solution NMR has a number of methods to measure the thermodynamics of the exchange more carefully in terms of kinetics or kinetics of exchange, the population distribution, etc. But I suppose that is not an option in this particular preparation because the molecular assembly is too large in this preparation.
No. I mean, for example, certain residues are not visible. Some are very broad because of heterogeneity. Some are they are also features of dynamics happening. For example, the alpha-synuclein, when the underlying 138B is added, there's duplication of certain resonances, and so that means for some slow exchange and so on. Also, for example, we do not know yet the stoichiometry of the alpha-synuclein oligomer in the presence of 138B. It could be different. So that might be also a point that we have to learn about. So that's no, I mean, but the dynamics maybe not as detailed as we can do it in solution, is also reflected in the solid-state NMR spectra. Yeah.
So without the molecule NL38B, it's a tetramer,
Yeah.
the oligomer.
The alpha-synuclein in
The
this
alpha-synuclein is a
lipidic
tetramer.
alpha-synuclein oligomer is a tetramer, and we think it is a. I mean, we have tried to extend it with the same conformation, and then there's steric clashes which prevent it from growing.
Okay.
So that might be the reason why it has a certain stability in the absence of monomers. Probably when monomers are approaching, then it does a refolding to a in register parallel arrangement, and then it can grow.
Okay.
And I mean, in the presence of monomers, it grows at a rate of hours, and hours, of course, can include very high barriers. And for example, the reorganization of an antiparallel beta strand to a parallel one means breaking of several hydrogen bonds. So that could be a very slow process, but it could be in line with what we see experimentally for this interconversion from intermediate 1 to intermediate 2, and then finally growing to the fibril. So that's.
How is this picture different in amyloid beta, A beta?
That we have no structure yet of the oligomer.
Okay.
We have the structure of the fibril, but not of the oligomer. So that is hopefully coming within this year, but we don't know yet. No, we cannot.
Is it true that other than humans, like chimpanzees, for example, probably do not develop the neurodegenerative diseases like Alzheimer's or Parkinson's, or that is not identified yet?
I do not know.
Okay.
Maybe I should know. I mean, it could be that really the human monkeys, like chimpanzee, orangutan, gorilla, also that, I mean, they grow old also, and so maybe they develop it. But I really have no idea. I mean, what I know about type 2 diabetes, that is something that certain species develop, like, for example, cats.
Really?
Cats can be diabetic. I mean, there's, for example, in Munich, there's a clinic for diabetic cats.
Okay.
So, I mean, probably what one does is put them on a diet or so, and maybe give them also the medication that human beings are getting. Well, for example, rodents don't develop it. Now, one could say, I mean, the rodents live not so long. but they also have a IAPP which is not aggregating, and so it cannot produce what. But well, the cat IAPP and also monkeys can also have diabetes.
Okay.
That can aggregate. So that's at least currently one of the reasons why people think, okay, IAPP is not just an epiphenomenon in diabetes, but that it is really kind of driving the disease, that those species that don't have aggregating IAPP also don't have diabetes, while those that have aggregating IAPP also develop diabetes under this more or less the same conditions as human beings, and especially cats, which are pets.
Right.
I mean, they're overfed by whatever the owner, and then they develop diabetes.
So the pet can also get lifestyle disease if the
course,
owner also.
course. I mean, some of these cats are humongous, yeah.
Right,
So,
right.
and it's not just the fur.
Right. I had one thing I was wondering for the molecule NL138B. So it has to cross the blood-brain barrier if
Yeah.
it's to be effective
Yeah.
in the Alzheimer's or
Yeah.
Parkinson's disease, possibly, especially if it's effective for Alzheimer's, and it does cross the blood-brain barrier.
It does. I mean, the concentration that that we find in the blood compared to the brain is blood 1,
Okay.
brain 5 to 10. So the concentration in the brain is higher than in the blood. So
Really?
in principle, it's not a barrier for this type of molecule, and that's due to the fact that it's pretty lipophilic. The bigger problem was, before we had a nice formulation, which is now also there and is published, was to go over the gut-blood brain blood barrier, and with the right formulation, and also with a prodrug, which is kind of yet another formulation, it is possible to reach quite, I mean, sufficient levels such that the N138B is now in this phase 2 trial, and we are going to test the prodrug also very soon in patients in order to see how it is resolved there, and that's the so uptake is not an issue.
Okay. I had one question regarding the anti-diabetic properties of the molecule, and that is preventing the IAPP aggregation. Is recently, I think we have seen that there is a lot of glucagon mimicking molecules, the GLP-1, which are very popular anti-diabetic drug, but it also turns out to be anti-obesity drug and weight reduction molecule. The likes of Ozempic and Wegovy and other versions of which are with the market now. So I guess it's a two-part question, that does these class of molecules that you described, which are preventing the IAPP aggregation, does it also have like anti-obesity properties or
No?
Okay.
At least, I mean, we have no idea, of course, in humans, because
Right.
it's not applied in humans. I should say so far. Hopefully, at some point in time, it will be. But we saw in the mouse model that the treated IAPP, I mean, the treated mice which were obese in order to induce type 2 diabetes and were expressing the IAPP, that those actually were heavier, the treated ones were heavier than the untreated ones.
Okay.
And one of the reasons is that, I mean, these mice eat as much as they can.
Right.
And apparently they, I mean, they when they're healthy and really functional, they eat as much as they can, and they apparently, the treated ones, eat more than the untreated ones. So yeah, that's the finding. So it's clearly not an obesity drug. Regarding these drugs, I mean, I have no idea. Fortunately, I'm not too heavy, such that I would be exposed. But I recently had a meeting with a former actually basic school mate whose father was already pretty heavy, and he also is heavy, but also quite muscular. Yeah? And but still, I mean, he tried this molecule out,
Okay.
and he said after some time he stopped doing it because it apparently is reducing the appetite quite a bit, such that I mean he talked about one incident where he went to a restaurant and he ordered his favorite meal, and then the favorite meal came, and I mean he ate a few bites or so, and then he was finished. And then the waitress who was serving him apparently many times, she said, "Oh, was something wrong or so? Did you not like it?" And he said, "No, I mean he just doesn't even like to eat his most favorite food."
Right.
So in principle, it's a killer of any joy
Right.
in life.
Right.
Also, he mentioned, I mean, a glass of wine or so, yeah,
Right.
which he also likes to drink. Yeah, I
Right.
mean, he said he could not enjoy anymore. Yeah.
Right.
And so then his conclusion was, okay, I mean, I rather try to eat less, but with joy, than
Right.
to eat these drugs. Whether it is a singular observation or whether everybody feels that, I have no idea. But of course, yeah, extending life at the cost of one of the enjoyable activities, namely eating, is of course also a misery. Yeah.
Maybe there is like a law of conservation of joy that if the total joy is conserved, if one extends the life, the intensity of joy will reduce.
Yeah, I don't know, but I mean these molecules are, I mean, at least from this one observation, seem to have these effects. And there's also the consideration that they might whatever do something to the pancreas as well, and so that's also something that probably only when one has this molecule in the market for a longer time, one can really
Yeah,
estimate
that's
whether.
exactly what I was thinking, that probably it's too short to know about the side effects
Exactly.
which might appear maybe 5 years of usage or so.
Exactly.
So,
Yeah.
right. Now, there is always that danger, which is it seems like these molecules are almost too good to be true for.
Yeah. Well, I mean, it has these side effects of being a joy killer,
Right.
and of course, it's a commercial, absolute commercial success. Yeah.
Yes,
And
absolutely. Yeah.
it seems many people need it, and if one looks also now in India, I mean,
Yes.
it's quite amazing how many young people are there which are clearly overweight.
Right.
And I mean, this pandemic of obesity is clearly there, and somehow has to be dealt with.
Right. What is the so I know that one of the biggest possible thing that you are looking forward is the clinical trial of these molecules, but also overall, I want
Kind
to
of the
ask
result of the clinical trial.
result of the clinical trials. Uh, so was wondering that if you are thinking of what are the next motivating challenges, one is overall the impact of this program or scientifically that you are
Well, I
looking
mean,
forward to.
in principle, the mechanism of the activity of this molecule is interesting in itself because it has some activity in mice. Yeah? So at least one would understand what is happening in mice. Whether the same effects happen in humans, so whether one can translate from mice to humans, in principle, with all these diseases, nobody knows.
Right.
So, but that's another question, and that question can be answered with the outcome of the clinical trial. If there's little or no effect or so, then apparently the mechanisms that one tries with the mouse models to recapitulate from the human situation are not recapitulated.
Right.
And then at least one has the insight, okay, this, whatever, aggregation or interference at this level or so seems not to be the right target.
Right.
On the other hand, if one sees an effect, it would show, okay, there's some translatability from mouse to humans, and then most probably the biochemical effects and the structural biological effects that we see would then probably also have some importance in the human situation. Yeah. So both outcomes are, from a scientific point of view, nice. Of course, the nicer result would be that all the people, and I'm not one, all the people that have designed those mouse models in order to recapitulate these diseases have done it correctly, yeah, or not correctly. I mean that they, yeah, that they were designed in such a way that they really recapitulate the disease. And I mean, a little hope actually comes from the success of the Alzheimer antibodies, lecanemab and the other one, gentamab or so. They show a slowing down by about 30% or so of Alzheimer cognition decline. And they have, in principle, the same target as we have. Such antibodies have so far failed with other targets, with tau or with A-beta, with tau or with alpha-synuclein. And one reason one could imagine is that tau and alpha-synuclein are aggregated within the cell, while A-beta, a large portion of A-beta, is outside of the cell. So it can be, with an antibody, reached more easily, while the other proteins are less easily reached, because the antibody not only has to go through the blood-brain barrier, but also then from the extracellular space to the intracellular space. Yeah. And that could lead to concentrations that are just not enough. But the fact that humans slow down cognitive decline under the treatment of the antibodies seems to indicate to me that the target, namely aggregation, is a valid target. And one just has to apply enough of the molecule such that this aggregation is impeded or modified even stronger than with the antibody. And then one would expect, with the knowledge that one has now, that there should be a reasonably strong effect. How strong it will be then in the human situation, the various diseases, this is exactly what this experiment, the clinical trial, will find out. To now make any prediction would be very difficult. For the disease that it is running now is this multiple system atrophy, where there's no treatment at all available. So let's assume one would delay the progress by 50% or maybe 100%, meaning, slow down by a factor of 2 or so. That would be a huge success for that molecule, but also then, of course, for the patient. Some cancer medications are admitted for the treatment, which extend the lifespan of human beings by half a year or so. Yeah. So that's any time one can gain under a treatment is, of course, super valuable to the patient and also the families of the patient. And so that's one has to see whether there's an effect. Of course, everybody hopes it's as big as it can be.
This is, of course, at the cutting edge of structural biology and biophysics. And recently, the artificial intelligence programs and AlphaFold has made quite a big splash in this field and made some deep impacts. I was wondering, what's your perspective? How has it changed the research program, helped, made it faster, or how has it impacted this kind of the projects that you have described?
We are mainly working with intrinsically disordered proteins, where the experimental basis on which one could train AI is just not big enough in order to use it. I mean, one has to remember that AlphaFold is based on 80 years of crystallography with hundred thousands of structures, and NMR spectroscopy has also contributed something like 10% or so of the structures, and now also cryo-EM. So without this large basis, which is experimental, there would be no AlphaFold. And of course, by including now structural ensembles of intrinsically disordered proteins, one can then also think of training there. And then probably at some point in time, there will be also contributions from artificial intelligence there. Then an additional complication is we work with membrane proteins, and membranes are still apparently difficult with AlphaFold. So in principle, we are working with species where experiment is needed, but with the deposition of these structures, then we will also allow in future years to come AI to get the best out of such structures here. But at the moment, I would say the importance of artificial intelligence regarding the structures is not there. I mean, what is coming more and more is AI in processing of NMR spectra and so on. So that of course we use and take into account, but that one would substitute, for example, where does the small molecule bind in the structure, and how does the structure look like? This is at the moment purely experimental. Of course, I mean experimental together with the kind of conventional tools of structure determination, namely molecular dynamic simulations and taking restraints into account and so on. Yeah, but it's still a pretty conventional methodology. Also, cryo-electron microscopy, the particles are too small, probably also too heterogeneous in their structure. X-ray crystallography is also not possible. So here really for atomic resolution structure information, NMR is the only tool. That in principle, I mean, of course, we because we have NMR instruments, and fortunately such projects are of some potential medical interest, and they can only be solved with this method. That's why they are of interest to us. And I mean, I was once in an evaluation of a NMR spectrometer initiative where an X-ray person whose name I don't want to tell now, but during the evaluation, then he said, I chose in my career only projects that didn't need NMR, and I could do everything with X-ray. So, I mean, here we choose projects where NMR can make the biggest contribution. Yeah. So, and we have the freedom of science, so we can choose whatever we do, whatever we choose. And I think first of all, from the novelty of the structures, and also from the at least the potential importance of the structures, I think also from kind of the if one looked at this with the applied point of view, one would probably say, okay, why don't you do it? No.
I really like the part where you said NMR is the only tool for a big class of problems.
Yeah, I mean, IDPs are really, I mean, the number seems to be increasing. I mean, some years ago it was 30%. Now it seems, I mean, sometimes I read 40% of the sequences that are expressed in higher organisms are intrinsically disordered. And therefore, atomic resolution, structural insight, or ensembles and so on, NMR is the only tool. Another big topic is phase separation, which also most of the time is done by IDPs. And that's also something where cryo-EM just sees blur, X-ray doesn't see anything. I mean, one can do fluorescence and stuff like that, but this is then kind of certain specific points where one gets information. But that's there also. It is the only technique which delivers information, and so in kind of two big classes of, and this is just proteins,
Right.
NMR can uniquely contribute. And also in the next future, I think, at least the experimental characterization of these systems is necessary to train AI models. So at some point in time, there will probably also AI then making better and better predictions. But the experimental basis has to be bigger. And so principle, I always say, I mean, the Nobel Prize with AI just shows how good the decisions of previous funders were to invest in synchrotrons and in NMR machines, and now also in cryo-electron microscopes to allow these AI models to be trained. So the reaction should be rather invest more in experimental science than to think without investing in that AI will also solve the other problems. No, it will not, because it needs the experimental basis for being trained.
Fascinating. And thank you so much for your time and for this wonderful conversation. And thank you. And we'll be back with a new episode soon.